15-minute or less lecture series, Anatomy and Physiology, Chapter 10, The Senses, Part 1. Sensations are conscious or unconscious subconscious changes that we are aware of. It includes a stimulus, which is the change in the environment, a sensory receptor that detects the change and transmits it to the sensory neuron that conducts the impulse to the brain where the information is integrated. Sometimes the sensory receptor can be the sensory neuron. There are classifications of sensations. There are the general senses, which include the somatic senses, things associated with the skin, visceral sensations associated with our internal organs, and the special senses that are localized to the head, such as smelling, hearing, taste, vision, and equilibrium. The types of receptors out there can be defined by what they detect. So photoreceptors detect light. Mechanoreceptors detect a physical change to the receptor itself. It's somehow being pushed or bent, or otherwise something physical is happening to it. There are thermoreceptors for temperature change, chemoreceptors for different chemicals, especially say chemicals in the composition of the blood, and of course pain receptors, which can detect many different kinds of stimuli. Sensory adaptation is basically just meaning that the brain must prioritize what's coming in it. It's getting a lot of sensory information, so it needs to decide what is most important. And so sometimes the brain will become less responsive to a stimulus that's occurring over a long time. So for instance, if you applied, say, cologne in the morning, later on in the day, you might think that the odor has worn off, but actually it's still there. It's just that your brain has become desensitized to that sensation. General senses. General senses are associated with the skin primarily, but also could include muscles, joints, and organs in the body. Many of the sensory receptors are also the sensory nerves, and it is the dendrites of the nerves that are acting as the receptor. They are commonly associated with epithelial tissue, especially with the skin, and can detect things like itching, change in temperature, pain. A specific example is the tactile corpuscles. These are where the dendrites of the neurons are covered in sheets of dense connective tissue, and they are sensitive to touch. So that if something pushes on your skin, it'll change the shape of the tactile corpuscles, leading to a signal. They're also deeper in the dermis of the skin, the laminated corpuscles. These are also, again, the dendrites of sensory neurons wrapped around by connective tissue. And in this case, they are detecting pressure, which is a little bit different from touch. So with pressure, they can determine how hard something is touching you rather than just if you're being touched or not. The various temperature sensations are also detected by free nerve endings. However, these are not wrapped around by anything. There are two main kinds, the warm receptors that work from 25 degrees Celsius to 45 degrees Celsius, and the cold receptors from 10 degrees Celsius to 20 degrees Celsius. These are both rapidly adapting, so they will stop sending you information about the temperature rather quickly. And also worth pointing out that extreme temperatures, extremely cold or extremely hot, send the sensation of pain rather than these thermal sensations. Pain is also detected by free nerve endings, and the sensation of pain can be caused by many different stimuli, such as tissue damage and the chemicals released from that damage, from oxygen deprivation, and from extreme stimulus. So, for instance, if it's really dark, so you wake up in the middle of the night, and you turn the lights on and it hurts a little bit in your eyes, that's an example of extreme stimuli causing pain. Now, overall, pain is a good thing. It's letting us know when something bad is happening to our body. However, it can be a problem if the pain is chronic and persists for a very long time and is unable to be resolved. It's also worth pointing out that pain receptors are found everywhere in the body except the brain. The brain is the only organ that does not have pain sensation. Referred pain is when the pain is associated with a body part that's different from where the actual issue is occurring. So for instance, the most common example would be a heart attack. Someone's having a heart attack, the damage, the issues with the heart. However, they often report feeling shooting pains in the left arm, even though obviously the left arm is not where the heart is. It is thought that referred pain is actually The brain getting a little confused from signals arriving from a common nerve pathway, so both the left arm and the heart. Pain fibers can be acute, which means they only send signals quickly and for a brief period of time, or chronic, where they persist with a slow, dull sensation over time. Um, Pain impulses are initially processed in the spinal cord if it goes through the spinal cord. However, it is not until it reaches the thalamus that you actually start to get a really conscious idea of the pain and where it is in general. And then the cerebral cortex really helps to let you understand where the pain is and how severe it is. So again, you become aware of the pain when it reaches the thalamus, but it is the cerebral cortex that decides on the location, intensity, and what your response should be. And that could be an emotional response. Often when we're little children, our emotional response to pain is to cry. As adults, at least for me, I tend to get angry when I feel pain. The brain also tries to limit the pain to help control it by releasing endorphins to help act as a natural control. All right, moving on to the special senses. We'll start with olfaction, or the sense of smell. It all starts in the roof of the nasal cavity in a structure called the olfactory epithelium. Here you'll find olfactory receptors. These are the neurons that are able to bind chemicals. They have these hair-like cilia that project into the mucus produced by the mucous membrane of the nasal cavity. So chemicals that dissolve into the mucus bind to the cilia, to the hair-like structures of the sensory neurons. That binding is what leads to a signal. So that makes the olfactory receptors chemoreceptors. They then transmit the information through the skull and into the olfactory bulb. Then the olfactory bulb transmits that signal directly to the primary olfactory area in the temporal lobe. The thalamus is completely bypassed. So it goes from the receptor sensation to the olfactory bulb to the temporal lobe. No thalamus involved. There are thousands of different chemicals that can bind to the sensory neurons for olfaction. There are 7 that are common to everybody. The other thousands can vary from person to person, which is why some people like some smells and other people like others. Basically, the way it works is when the chemicals bind to the sensory neurons, they open up sodium ion channels, then the sodium floods into the cells, causing the generation of the action potential. And as I already mentioned, olfaction is one of those sensations that adapt quickly so that you can get used to a smell. Gustation. Gustation is the sense of taste, localized to primarily the tongue. There are these bumps called taste buds scattered all over the anterior surface and the superior surface of the tongue. These bumps are called papillae. There's also a few on the back of the throat, the pharynx. If you look at a taste bud, you see that there are taste cells. These are modified epithelial cells that are acting as the receptors. They have little hairs that push up to the surface of the tongue and will bind to chemicals that dissolve in water, aka in the saliva. So the chemicals that we can detect have to be able to bind in water and then bind to the taste cells. If enough chemicals bind, then the taste cells will pass that information, the signal, on to the sensory neurons. And so that means, of course, that the taste cells are chemoreceptors. There are 5 main types of taste: sweet, sour, salty, bitter, and umami. Turns out that maybe there's some more. They're still working on research on that, but these are the main 5 ones. They're localized to different areas on the tongue. Umami, by the way, is the sensation of meatiness, and it is actually signaled by a derivative of amino acid called monosodium glutamate. The taste sensation goes through the facial nerve for the anterior 2/3 of the tongue and the glossopharyngeal nerve, the posterior 1/3, through the thalamus and to the primary gustatory area in the insula. Hearing and equilibrium. It turns out hearing and equilibrium are localized to the internal ear. The outer or external ear consists of the auricle, the big fleshy part we can touch. The sound waves get funneled into the external acoustic meatus, and they travel down to the tympanic membrane. Here's the tympanic membrane, which then vibrates when the sound waves beat against it. Then we have the middle ear, that is an air-filled chamber after the tympanic membrane. Here we find the auditory ossicles. These are 3 bones. the malleus that contacts the tympanic membrane, the incus, and then the stapes that fits into the oval window leading to the inner ear. As the tympanic membrane vibrates, it causes the malleus to vibrate, which causes the incus to vibrate, which causes the stapes to vibrate, which causes the transmission of the vibration to the inner ear. In the middle ear, there's also an auditory tube that connects to the pharynx. This allows for air pressure on both sides of the tympanic membrane to be equalized. Often experience this rapidly in, say, airplane flights where you get that popping sensation in the ears. The internal ear is made up of the outer osseous or bony labyrinth, and that is a hard structure, and inside that is a membrane structure referred to as the membranous labyrinth. There is fluids both outside of the membranous labyrinth called perilymph and inside it called endolymph. The inner ear is broken down into three regions. You have the semicircular canals, these little curved structures. You have the vestibule, the center area, and then the cochlea, the snail shell structure. The cochlea houses the organs for hearing. Semicircular canals and vestibule houses these structures for equilibrium. Within the cochlea, we see that we have the oval window again. When the oval window fits with the stapes bone, when the bone vibrates, it leads to pressure waves in the fluid found in the upper chamber, the scala vestibuli. The vibrations travel along that, will eventually pass through the cochlear membrane and into the scala tympani, which then will carry the remaining vibrations out the round window. The cochlear duct is found between the scala vestibuli and scala tympani. This is where we'll find the spiral organ, the actual structure that detects sound. If you look at a close-up of the spiral organ, you see that we have this tectoral membrane, this cartilaginous structure, and these little bitty hair cells. They have little hair-like projections that are embedded in the tectoral membrane. So when the pressure waves in the fluid hit the tectoral membrane, it will vibrate, bending the hairs on the hair cells, and this is what generates the signal that gets passed on to the sensory neurons. So the hair cells are a type of mechanoreceptor. So the way it works for hearing is we have the sound waves arrive in the external acoustic meatus. They bounce into the tympanic membrane, causing it to vibrate, which leads to the malleus to vibrate, which leads to the incus to vibrate, which leads to the stapes to vibrate, which then causes the transmission of the vibration into the fluid found in the scala vestibuli. Those pressure waves in the fluid will travel down the cochlea until they reach the area that is designed for that frequency. It will pass through into the cochlear duct, hit the spiral organ, the tectoral membrane of the spiral organ. This will cause it to vibrate, activating the hair cells, generating the sensation of hearing. And then the remaining pressure waves will travel through the scala tympani down to the round window where they will be dissipated. It turns out that the cochlear membrane, this entire structure, has frequencies that is attuned to. So that high frequencies will enter early on, middle frequencies will travel further down, and the low frequencies are at the very end of this membranous structure. So basically, our cochlea is sort of like a piano with different frequencies at different regions. Finally, the hearing pathway. So the signals pass through the cochlear branch into the vestibulocochlear nerve. That will then, of course, go to the brainstem and be transmitted up into the thalamus, where then it will be transmitted to the primary auditory cortex, which is found in the temporal lobe. It is worth pointing out that the sounds you hear in your right ear get sent to the left side of your brain and vice versa. That is it for this lecture. Be sure to subscribe and like.